Bevan Weir from Landcare Research, and Bevan's going to be telling us about pathogens, genomics and biosecurity. Thanks, Bevan.
G'day. So I am a research group leader for the mycology and bacteriology team, and the entire rest of my team is currently in Northland for the fungal foray, which happens every year, and it's kind of turned into a myrtle rust surveillance foray at the moment. So why are we doing pathogen genomes? I mean, I've got a long statement here, but it's kind of obvious, right? Bad things come in. So how are genomics useful for dealing with these nasty pathogens? Many years ago, maybe about nearly 7 years ago, I did my first genome in this space, and it was on a big one. It was on the Psa disease of kiwi fruit. So this came in about November 2010, and the diagnostic PCR test at the time picked it up as PSA. But there was a question, was it what was at the time called the Asian strain or the Italian strain? And PSA was described from Japan, but the strain that was found in Italy was much, much more virulent. So we did some genomes, and there was a good technician, Dukshul Park, there that did the work, and analysed it. Now, my problem was I didn't have any reference genome, so all I had was some sort of multi-gene work. And so I simply looked through the genome and found that indeed we had some differences there. So these are effector genes, which are kind of a pathogenicity genes. And so HOP-A1 was missing in the Asian strain, but present in the Italian and the New Zealand strain. And unfortunately, it's these toxins, interestingly, present in the sort of less pathogenic strain, but non-present in the Italian strain. So yes, unfortunately, I was able to let MPI know that very likely we did indeed have the Italian strain, which is now called PSA-V. So immediately, this is a great use of genomics in that we can very, very quickly get access and sequences of every single gene in the genome and compare them. So it's really just multi-gene analysis, but at the next level and much, much faster, especially now. And so we've got this organism, Pseudomonas syringae, is the species, and the pathovar, or what it's pathogenic to, is Actinidiae. And this system was actually developed in New Zealand by bacteriologists here in the 1980s because they couldn't distinguish Pseudomonas species from each other with the tests at the time, but they did know that they were pathogenic on different hosts. And now we've got another level as well that are revealed by genomics, and these sort of strain level differences. Of course, that was 7 years ago, and now things are much easier. And so I did a paper last year when we released 62 genomes from all of the pathotype strains of Pseudomonas syringae. And these are all freely available and out there. And the great thing now is we're much, much more prepared for the future should a new Pseudomonas syringae disease come in. We've got a whole bunch of reference genomes that we can compare to. One of the other nice things about genomics is that they can reveal function. So a few years after this PSA disease, there was a problem with a fish pathogen causing gleasing on a certain kind of fish. Now, diagnostic tests indicated that this could be a possible serious pathogenic species, but it was motile, so it was kind of swimming around, and the reference strain, the pathogenic strain, wasn't motile. Why? So again, we did the genome, and I did a whole bunch of things, looked at a whole lot of genes and toxins and various other things, but in this particular example, I was just looking at these lateral flagella genes. So the flagella are these whippy things on the outside of the bacteria that make them move. And so I could find that in the reference genome of the pathogenic strain, it had an insertion in the gene that constructs these flagella. But in our strain that we got from the fish here, it had an intact gene, so it was able to produce these flagella and swim around. Now, the astute of you might think, well, hang on, so you're saying that the pathogen doesn't have the flagella and it can't swim, but the wild type can swim around quite easily. And yes, so the pathogen has a bit of a disadvantage in that it can't kind of actively swim towards its host. But these sort of wailing, flapping flagella on the outside of the cell are a brilliant immune system target. So by making this gene non-functional and by not having these flagella, it's able to avoid one of the major things of host immune system recognition. Now, this slide probably should be called novel pathogen discovery, but I put the two options to my 5-year-old daughter, and yeah, she chose crusty bum disease. Its proper name is extrudative colitis or something like that. I just love crusty bum disease. Now, we all know that Kakapo endangered, and I know there's 154 birds left because I checked Wikipedia last night, so that must be the correct number. The problem with this disease is because we've got so few of these birds left, none of the birds have died from this disease yet, but potentially they well could if it's left untreated. And there are certainly implications for infertility. The thought floating around is that perhaps this was caused by a virus, but really it's a disease of completely unknown etiology. So, and I should say that I didn't do any of this work, but some of my collaborators, Daniel White at Landcare Research and ESR did this. So they took metagenomic swabs of the crusty bums of, a population of healthy birds, and then one particular individual before and after treatment with antibiotics. The results, well, the strongest sort of association there that the infected bird had, the big difference was a particular E. coli bacteriophage. And so bacteriophages are viruses that infect bacteria. And a bird is in the bacteria, so, it wasn't directly infecting the bird, but this bacteriophage is associated with a E. coli disease in human that causes great sort of gastrointestinal upset. But this was only sort of one sample, and we constructed specific primers for this bacteriophage and tested another bird, and we couldn't find it. So we need to do a lot more work here and collect more metagenomics. I'm fortunate enough to have a project funded by the National Science Challenge, and it's called Project 1.2, or in the long form, Application of Genomics and Metagenomics, a fast-track risk-based analysis of novel pathogens. Let's just call it Project 1.2. So the scenario is that we've got a bunch of novel organisms that we find in a group of known pathogens, let's say in a native forest. Now, DNA barcoding can give us great taxonomic information, but if it's something unknown, a new species, how do we know what significance this is? So the idea here is that we can directly characterize these pathogenicity genes, which we call pathogenic genetic elements in a broader sense. Our case study is going to be Phytophthora for intensely practical reasons. True fungi are much, much more difficult in terms of bioinformatics. And with Phytophthora, for example, one group of these genes are called RXLRs, and quite simply, every single one of them has this amino acid code RXLR in the sequence, so you can find them much more easily. And of course, Phytophthora are also, significant pathogens and economic and ecosystem impact. Very briefly, we're going to select, and this is sort of just starting over the past couple of months, we're going to select a bunch of different species representing different pathogenicity and ecosystem hosts and characterize these pathogenic genes and also novel viruses in them. Develop databases of associations and detect and track these PGEs using metagenomics. And Steve Wakeland, who gave a talk a few minutes ago, will be doing this part of the work. So quite a big project. Now, I think it's really important that we get more Māori researchers into biosecurity and genomics. There's just too few of them, too few of them in science in general, right? And my research group in mycology has had a program for the past sort of 5 years using summer students and getting them in really early. And I think this early intervention is important. And 2 of those summer students have come back, and one did a PhD, and now one of them is even employed as a technician in my group. So we get in early and recruit these people, and just let them know that genomics and biosecurity is a viable career pathway. So, in summary, the field of sort of pathogen genomics helps New Zealand's bioheritage, though by strain discrimination and tracking evolution. We had a talk by Nigel French this morning about that. Novel pathogen discovery, analysis of function, what these genes actually do, and potentially targets for plant breeding resistance. For example, in these Phytophthora, these RXLRs are specifically associated with plant genes. So the big idea, sequence all the genomes Now, is it really that big an idea? Because people are going to do these big pathogens anyway, right? But I think we need to do more than just the pathogens, but also lots of things that are just out there in the environment as well, as reference kind of sequences. And, Diane just gave a slide and showed the little nanopore sequencer. Well, I actually have one in my pocket while I've been speaking. And they really are this tiny and portable and relatively easy to use. And so sequencing is getting faster, cheaper, better, more portable. And I'll throw in a little bit about the myrtle rust that's, of significance at the moment. And there's an interesting story in beech forests. So in beech forests, there's Phytophthora diseases that are present, but they don't cause significant disease. But if you take a beech seed, take it out, grow it in a nursery, and then pop that plant back into the native forest, it'll die from Phytophthora. Now why? So it's got that same host genetic diversity out there, but the key difference is that it hasn't accumulated those sort of endophytic fungal complement. And so we really need, and particularly thinking about this in terms of myrtle rust as well, we really need to sample what is inside these myrtaceous plants, and particularly if they're showing any kind of variation in pathogenic response to myrtle rust. And we also need genomes of all these endophytic fungi as well to determine what the potential function of these things could be as well. So thank you.